are still widely used, not only for treating cancer, but also for treating other diseases, and also used for other things, let's say. Okay. Ending pregnancies, for example. Okay, so remember the medium that is used for the production of monoclonal antibodies, HAP medium, hypoxanthine, aminoterrin, thymidine. Okay. You have to provide hypoxanthine so that you can utilize the salvage pathway for the production of purine nucleotides, and you have to provide thymidylate because you can't synthesize thymidylate in this medium because the hydrofolate reductase isn't working anymore. Okay, and that's why you have these three components in HAP medium: hypoxanthine, aminoterrin, and thymidine. Okay? So, let's take a quick look at the degradation of nucleotides, and we'll start with purine nucleotides, which is relatively straightforward. So, when we look at the degradation of nucleotides, remember again what a nucleotide is. It contains a nitrogenous base, a sugar, and a phosphate. So if we want to degrade this, and we degrade it actually to produce, at least we do that, uric acid, purine structure, okay, we have to remove at one point sooner or later the phosphate from our nucleotide, and we also have to remove the sugar. Okay, so if we look at a pathway like this, we start out with a nucleotide and we produce just this purine structure here. We shouldn't be surprised if we have enzymes that cleave off phosphates and cleave off sugars. So if we start with GMP, for example, we first remove our phosphate here with the help of so-called nucleotidase, okay, just hydrolysis reaction to produce the corresponding nucleoside, guanosine, which just has guanine linked to ribose, okay. Then a nucleosidase hydrolyzes our N-glycosidic bond between guanine and ribose. We have guanine here, and then actually a deaminase removes the nitrogen here and introduces hydroxyl group to produce xanthine. So this nitrogenous base here is called xanthine, and this can be further oxidized with the help of an enzyme called xanthine oxidase to produce uric acid. Okay, so if we look at the degradation of AMP, we see that, okay, again, first we remove our phosphate, then we cleave off, no, actually, we first remove our nitrogen here with the help of a deaminase and convert our nitrogen that we would have in adenine here into an oxyl group to produce hypoxanthine. First, we produce a nucleoside. We have to remove ribose by hydrolyzing the N-glycosidic bond, and then we produce hypoxanthine. We oxidize this, utilizing the same enzyme as we saw here for this reaction, xanthine oxidase. We introduce first a hydroxyl group here, okay, and then further convert xanthine into uric acid. And uric acid is what we, the primates, actually produce and release as a degradation product, degradation of purine nucleotides. Okay, remember when we talked about the synthesis of de novo synthesis of purine nucleotides, I told you that birds actually secrete uric acid as a degradation product of degradation of purine nucleotides, but also to release excess nitrogen that comes from degradation of proteins. We know that we secrete excess nitrogen from degradation of proteins as urea, but we also produce uric acid because that's a degradation product of purine nucleotide degradation. Okay, other animals or other organisms, let's say, actually release nitrogen the nucleotide degradation products in different forms also. So we, primates, birds, reptiles and insects, we all produce uric acid. But then most other mammals actually produce something called allantoin. And if you compare the structures here of uric acid and allantoin, we see that we cleave this bond to the nitrogen here and the carbon in our six-membered ring. This is done with the help of an oxidase, which also produces carbon dioxide. This is secreted, as I said, by most mammals, but I want to point out that this reaction also requires water. Okay? And if you look at all these different degradation products that are produced by these other organisms, we see that they all include water for hydrolysis reactions. Now, the reason you could say that in organisms like primates, uric acid is produced is to save water, okay, not to utilize too much water to get rid of these degradation products. But this comes as a price, as we'll see in a moment. But let's first talk about the other reactions that ultimately can lead to the production of ammonia that can be safely secreted by organisms that live in an aqueous environment, like marine invertebrates. But if we just look at the next step here, we see that hydrolysis actually at this point here, catalyzed by an enzyme allantoinase, can be utilized in bony fish to produce allantoate, okay? And then another hydrolysis here can release glyoxylate and produce urea, which can be produced in amphibians and cartilaginous fish. Okay, and then urea can further be converted by urease to carbon dioxide and ammonia in, as I said, marine invertebrates. And all these reactions going down from uric acid to ammonia require water, and we don't do that. Okay. However, there's a problem because uric acid isn't particularly water soluble. If in your body you have elevated concentrations of uric acid, you may actually have uric acid precipitate in the form of crystals that you can see here in this picture. Do you know what this can actually cause? Kidney stones. No, kidney stones are usually the result of too much oxalic acid. Ever heard of gout? Okay, probably not old enough to suffer from it, but it's not uncommon, especially in, let's say, Western societies where people eat too much meat. So it's contained in a lot in meat, red meat, but also in legumes, for example. And if you have a certain concentration of uric acid then in your blood, you may have the formation of these precipitates, these crystals, and this is particularly bad if this happens in joints, and usually first manifests itself as gout in the joints of your big toe, okay, and then you get rather painful inflammations, and this has been known for quite some time because this picture actually stems from 1799. It is called the gout, so this is pretty painful. I myself, I occasionally have problems with this joint, but I don't suffer from gout. It's another reason actually in my case why I have these inflammations, but I can tell you this can be quite painful, okay? And people who suffer from gout can also tell you that this is quite painful. However, gout can be treated relatively straightforward manner by inhibiting the enzyme that produces uric acid, namely xanthine oxidase. Okay, if we go back here, xanthine oxidase produces uric acid, it also produces xanthine. But if we actually knock out this enzyme, xanthine oxidase, we don't produce uric acid, we produce still hypoxanthine in the degradation of AMP and xanthine in degradation of GMP, but both of these molecules are quite a bit more water soluble and don't precipitate as easily as uric acid. So how do we knock out this enzyme? With a competitive inhibitor, it can bind to the active site of the enzyme, and this competitive inhibitor typically is allopyrinol, and if you compare the structures of allopyrinol and hypoxanthine, which is the substrate for xanthine oxidase, the only difference here is that we have changed the positions of this carbon and this nitrogen. Otherwise it's the same structure, but it can bind to the active site of the enzyme and inhibit it. And that's an easy enough fix. Okay, let's take a very quick look at the degradation of pyrimidines, and we discussed this just utilizing thymine as an example. If you look at other pyrimidine bases, the reactions are similar or analogous. So what happens here is that we first reduce our pyrimidine ring system with the help of dehydrogenase. So here's a dihydrothymine that is produced. Then we hydrolyze, cleave a bond in our ring system to produce a linear structure. Here in this case it's sort of beta-ureido, because we have this ureido group, isobutyrate. Hydrolysis then removes nitrogen in the bicarbonate and produce this compound, beta-amino isobutyrate, and then we can actually convert this into methyl-malonyl semi-aldehyde, which can be converted to methyl-malonyl-CoA and succinyl-CoA, which can be channeled to the citric acid cycle. So I don't expect you to know any details here, so it's just a couple of hydrolysis reactions that then produce something that can be channeled to the citric acid cycle. Okay, so with this we conclude our discussion of synthesis and degradation of nucleotides, and in a way also our discussion of metabolism. However, this is really a matter of taste and definition, whether you look at it this way or whether you say that we actually continue our discussion of metabolism and go in the direction of the metabolism and particularly the synthesis of proteins. So just as a reminder, I think this is really the last time that I'm showing this picture. What we discussed in the last couple of weeks was first catabolism, so great variety of pathways we have discussed and reactions that led to the degradation of nutrients, carbohydrates, fats, and proteins. And we saw that a variety of end products are being produced, and that we can produce energy carriers in the form, for example, of ATP, other nucleotides, and then also NADH, NADPH, FADH2, which can all be utilized then in anabolic reactions. And we've focused pretty much on synthesis of sugars. We talked about synthesis of fatty acids and lipids, and we also talked about more limited discussion, synthesis of amino acids, and we just talked about nitrogenous bases and nucleotides. And we also saw how polysaccharides can be produced, or lipids can be produced. What we haven't discussed so yet is the synthesis of proteins utilizing amino acids as starting materials, right? And we haven't talked about the synthesis of polymers of nucleotides, namely nucleic acids. So if you look at it from this angle, and this is exactly what we're going to discuss from now on, synthesis of nucleic acids and of proteins, we're simply continuing a discussion of metabolism, right? So this is, let's say, a biochemist's point of view, potentially. You hardly ever hear this. What you usually do in classes like this is that, okay, your teacher says, no, now we're done with metabolism, let's move on to molecular biology. Okay, molecular biology, you can say, is a subfield of biochemistry that deals exactly with the the chemistry, biochemistry of nucleic acids, and the synthesis of proteins. But strictly speaking, we're still continuing our discussion of metabolism. So we will, in the last couple of hours, actually focus on the synthesis of proteins, and just to do the same thing that I discussed, that I did for the other classes of biomolecules, let's remind ourselves of what functions proteins have. Okay, so we had discussed many examples of enzyme catalysis in this class. We just had a talk on Tuesday about the utilization of enzymes for industrial processes. So not only in individual cells in our organism, enzymes are of great importance, but also for industrial applications nowadays, and for hundreds of years, for example, for the production of wine and bread and beer. But other proteins store stuff, okay? Myoglobin stores oxygen, for example, or transport molecules. Hemoglobin, for example, transports oxygen, okay? There are many proteins that provide structural support, do mechanical work, muscle movement, for example, or regulate cellular biochemical processes. So here we can talk about signal transduction cascades, for example, hormone receptors and things like this. There are many that actually participate in signaling events. So again, protein, hormones, for example. And then the last category, let's say on this slide, is simply called mediators, and I just talked about the role of antibodies as mediators between foreign materials in a body and other components of the immune system that can remove this foreign material. Then all antibodies do is act as a mediator between these two things, the foreign material and other components of the immune system. So, as you may know already, the information for proteins is stored at the genetic level in the form of DNA, okay? And by the way, a lot of the material that we are going to discuss the last couple of hours may be familiar to you from other classes, cell bio classes, or whatever classes you may have taken. I would encourage you to still pay close attention, because we as biochemists try to understand life at the molecular level, so we may take, in some cases, somewhat closer look at things, or look at things from a different angle, okay? But a lot of things should be familiar to most of you. And we will talk about this biological information flow that ultimately leads from the level of DNA, genes, via messenger RNA to functional proteins. Okay, and this will be a very abridged discussion of these processes. Okay, so I just told you that the information for proteins resides in genes. Okay, so genes are simply segments of DNA, or some organisms also RNA, so for simplicity we will just assume it's DNA, okay, that encodes the information for a functional biological product. The functional biological product doesn't necessarily always have to be a protein, but it could also be an RNA. Okay? But for simplicity, we will usually just think, okay, it's a protein. So the total genetic material that we find in an organism, then we can refer to as a genome. Okay? And it's again either composed of DNA or RNA. I mentioned before that the DNA then comes in the form of chromosomes. And chromosomes, that's something that I really at one point had to comprehend, because I